Everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about biotechnology, and that is using all the things that we human beings have learned through the studies of molecular biology and applying those to improve health outcomes, to make discoveries. And we're going to talk about just a few methods that we can utilize now that we know about DNA and RNA, replication, transcription, translation. Hope you enjoy the video. So one of the very cool things that we can do is use this. You may have heard of it before. This is a thermocycler, and it's used in the process known as PCR, polymerase chain reaction. And what this thing can do is it can amplify DNA or RNA so that we can study it a little bit easier. Basically, what happens in PCR is you take some genetic material, again, it can be DNA or RNA, and you heat it up. And you heat it up so that you can separate the DNA. Essentially, what we're going to be doing is we're going to be replicating the DNA and making lots and lots of strands. And in fact, when we do this, we get an exponential increase in genetic information. And in order to do this, you have to heat the DNA up to 94 degrees Celsius. That's enough energy to separate the two strands. Next, we're going to bring that temperature down a little bit into the 50s, usually right around 52 to 54 degrees Celsius, and that's going to allow some primers that we've added to our mix that will target certain genes. Once they've bound, then we're going to warm it up again, usually around the low 70s or so degrees Celsius, and that's going to allow a special DNA polymerase called Taq polymerase. It's not from human beings, it's from an aquatic organism that can withstand those really high temperatures. And so that Taq polymerase is going to go through and it's going to replicate the DNA. This is a complete cycle. Then what's going to happen is in the next cycle, we're going to heat it up again and separate all the strands that we've just made, and we're going to keep copying them. And so usually PCR takes around 30 cycles or so. And then once we have those 30 cycles, we have enough DNA that we can actually study. What's even cooler, and I think at times more useful, is reverse transcription PCR, otherwise known as RT-PCR. Instead of working with DNA, what we're doing here is we're recognizing and amplifying RNA. And the reason why I think this is so cool is that it gives us an indication of how cells respond to certain environmental parameters. And so if we're doing experiments on cells and we want to see what genes are active at any given time, or at least that if the genes of interest are active at any given time, reverse PCR is a way to do this. Not only that, but with reverse PCR, reverse transcriptase PCR, you can also quantify the amount of RNA that's present. So you can not only just see whether or not certain genes are active at any given time, you can see how active they are. One of the things that we do with PCR is once we have enough DNA that is present, we will separate the DNA based on its overall size. We do that using gel electrophoresis. And you can see in this picture here, basically what we're doing is we've got this agarose gel, and we've got some wells, those little 3 areas right there at the top of the gel, and that's where we're going to add our sample of DNA. We're going to run a current, usually that gel is submerged in a buffer. We're going to run a current through that buffer and through that gel, and since DNA is negatively charged, it's going to be attracted to the charge that is opposite of it, which is the positive side. So the DNA is going to run through that gel from where the wells are at the top of the gel, at the negative end, towards the positive electrode. And what happens is this gel has little tiny microscopic pores. Now imagine you've got DNA of different sizes, and what's going to happen is the DNA that is smaller, it's more nimble, it's going to be able to get through those pores a little bit faster than the larger, slower, more cumbersome pieces of DNA. And so the smaller pieces are going to travel through the DNA, the gel electrophoresis faster, and there you can start to separate DNA based on size. This is a characteristic gel, and so what we're seeing here is those bands are a collection of DNA at a given size. And one of the things that's not shown here but is often very, very necessary is you can use what's known as a DNA ladder, and that DNA ladder will give you certain known sizes of DNA and how they separate. And then you can compare the DNA that you're separating in your specimens to the known sizes in your DNA ladder. This is also really useful for things like DNA fingerprinting. You may have heard that before. When a suspect leaves behind DNA at a crime scene, You can use the process that we've talked about here, the PCR and the gel electrophoresis, to match crime scene DNA with a particular suspect. Perhaps one of the most interesting things is sequencing DNA, and so we can sequence the genetic material of lots of different organisms, microbes, all the way to human beings. In fact, when we sequence the DNA of human beings, the entire genome, in the 1990s, it took almost 10 years, but we discovered a lot about living things by sequencing their genetic information. And in fact, we can make inferences based on genetic similarities. And so if certain microbes or yeast or even worms have a gene sequence that's similar to human beings, and we can figure out what it's doing in those other organisms, well, we can infer what it's doing in human beings. And one of the most popular ways to sequence large genomes is through the shotgun approach. And basically what you do is you chop up the DNA into different size pieces and you look for overlaps. And once you have enough of those overlaps, almost like parts to a puzzle, you begin to put the DNA together and get an entire sequence. So these concepts and these machines that we use to analyze DNA has been really important, because what we can do is then we can apply these things that we're learning about to genetic engineering, altering genetic information in some way. One potential way to apply what we've learned in molecular biology is through gene therapy, and that is when an organism or a human being has a genetic abnormality, there is a potential to replace that bad gene or at least stop it from functioning. And so here's a really good example of gene therapy. You've got a person who has a genetic disease. This one happens to be on the X gene, but in here we isolate those cells. And with this particular method, we're using viruses that are going to transfect the cells in vitro in a culture. And then those viruses are going to attach to the cells, gain entry. They're going to release hopefully the corrected version of a gene. Then we grow those cells up, reinsert them, and that is one way to actually accomplish gene therapy. But there are other ways besides this. Now, to really do gene therapy, it implies that we have to be able to insert a gene. I just showed you one way using viruses, but the problem with viruses is it's not super reliable. They don't always gain entry into the cells, and they don't always insert the genes where they're supposed to. And so we've come up with better ways to insert genes and do genetic engineering. One way is to use something called the restriction modification system. And what that is, it's two parts. It's actually derived from bacteria. Bacteria use a restriction modification system to protect themselves from viral infection. There's two parts, like I said. One are restriction enzymes. And restriction enzymes, think of them as like little pairs of scissors. And the reason why I have this picture here for you is this is a small stretch of genetic information. Now, these scissors are super specific. And what you're seeing here is that the scissors, the restriction enzymes, will cut any sequence where you see that AATT in this particular example. And you're going to see that the exact same sequence also exists on the second strand. And so it cuts often in a kind of a zigzag pattern, if you will. Those are called sticky ends. And those sticky ends, once we separate that, we're going to have AATT exposed on both of those edges. And that, once we know that, will allow us to insert genes. So here's what that looks like. Let's say you've got a plasmid. We've talked about plasmids before. These are extra chromosomal pieces of DNA found in bacteria, not necessary under normal conditions. And what we have here is a plasmid. We have a site that we want to augment, that we want to change. And so we're going to use the restriction enzymes because they recognize sequences that are close to the site that we want to change. And they're going to cut. Now you're going to see in the middle picture what they've done is they left those sticky ends. And since we know what the sequence is, because we know what restriction enzyme we're using, there's lots of different restriction enzymes. All of them have different sequences that they're going to recognize. So if we know the restriction enzyme we're using, we know that the sticky end sequence, we know what that's going to be. We create a piece of DNA that has a gene that we want to be expressed, and we insert that. And that's called recombinant DNA. We've recombined two pieces of DNA. So now we have this plasmid. So next thing is, okay, we've got this recombinant plasmid. We have a gene that we want. Well, maybe we want to produce something from that gene. And so we insert that plasmid into the bacterial cell in a process called transformation. Now what's going to happen here is once that bacterial cell has that plasmid, if we encourage it to grow, it is going to go through something called vertical gene transfer. And every single time that microbe replicates, it's going to replicate its chromosome, which is the big blue squiggly thing, but it's also going to replicate the plasmid that we've inserted. So now we've cloned these microbes. They have this gene of interest. Let's say this gene is producing a growth factor or insulin, and maybe we can do this much, much cheaper using bacteria than if we were to try to extract it from living things that are producing it. But you want to know something that's really cool? We have an even better way to insert genes, and that is via the CRISPR system. Now, the CRISPR system, again, this is a bacterial defense mechanism. It's used by microbes to protect themselves from viral infections. Well, we discovered it, and we discovered a way to make it useful for human beings. What's interesting about the CRISPR system is it's very, very specific regarding the locations of DNA that it will recognize. And so this is a big, complicated picture, and I'm not going to go through it step by step. But the summary version of it is we can take the CRISPR system, it can recognize a certain gene sequence, and it has the ability, through the use of the CRISPR system and restriction enzymes, to cut out a section of genetic material and potentially insert corrected versions of genes. And again, most importantly, do this very, very specifically. Minimally, it has a very specific recognition point. It cuts out that sequence, and even if it's not replaced, we may be able to knock out that gene, which is really important if the gene should not be functioning. And so, yes, we have ways to alter genetic information, but there's also other useful things that we can do, one of which is this concept known as metagenomics. Now, genomics is the study of genetic material. It's the study of where genes are located and how they function. Metagenomics is a little bit different because what we're doing here, it's primarily based in microbiology. And the reason for this important concept and this important technique is we can only culture about 1 to 2% of the microbes that we know to exist on the face of the earth. And microbes are foundational for food chains. They're, believe it or not, foundational for human health. They cycle nutrients. They have all kinds of advantages. And so it's to our advantage as human beings to understand what microbes are doing and where they are located and in what concentrations. But like I said, we can't grow a lot of them in lab, and so sometimes it's really hard to gain that information. Well, metagenomics is a little bit different because you don't have to necessarily grow the microbes to figure out what microbes are present in a particular sample, whether it's a soil sample, maybe it is aquatic substrate, maybe it's from the human body. And so we take a sample and we extract the DNA, and then on the right-hand side, we can identify the microbes based on their genetic information, their genetic sequence. But not only can we identify what microbes are present, we're getting better and better at identifying the ratio and the relationship between different microbes based on the amount of genetic information that's present. So collectively, we gain a lot of information, and one of the hottest areas of biology right now is bioinformatics. And if you are somebody who's really good at math and analyzing a lot of information, or you really love AI, bioinformatics might be for you because it is the study of biological data. It's the study of protein function and structure, really, and then from there, protein function. We're doing a lot of data mining, and so there's a lot of information out there that's already been published, and now it's just a matter of fact of putting all these different research papers together in a certain subject and coming up with meaningful data that's called meta-analysis, disease prediction, which can lead to drug discovery. And so there's so much out there in terms of even genetic information, the structure of DNA, epigenetics, which is the study of how genetic information is structured, but not necessarily its sequence information. And so what we're doing is we're learning, when we learn about molecular biology and about DNA and RNA and protein, eventually the point is to use that information to make discoveries, to figure out what genes are doing, to figure out what proteins are doing, to figure out how all these things relate, and use those to inform us on how to live a better life. One other really cool discovery that's out there is something called RNA interference. For the longest time, when we would sequence genetic information, we primarily looked at one main part. It was called an exon of our particular chromosomes. Exons are expressed. This is the part of DNA that is used to make proteins. But there's this whole other part that's called an intron, and these introns are not expressed to make proteins, and it turns out that they make up the majority of our genetic information, but they don't seem to have a purpose. At least they didn't seem to have a purpose for many decades. In fact, at one point, they were called junk DNA. Well, luckily, there were some smart scientists who kind of assumed, hey, if you've got that much DNA left over inside of cells that aren't coding for proteins, don't know if we should necessarily be calling it junk. It's probably got a purpose. And sure enough, they figured out that a lot of these introns that don't actually code for proteins are used to regulate the expression of RNA once the RNA leaves the nucleus. And so RNA is used as a recipe to make proteins. We say that RNA is translated into proteins. Well, it turns out that these introns, small parts of them, can leave the nucleus and bind to messenger RNA and shut it down. Now that we know this, we can use these small mirror images. It's based on base pairing rules, the A's and U's and C's and G's. We can use these introns to bind to messenger RNA, and that marks it for degradation, to be broken apart. And we can silence, in essence, we can silence those genes, but do it a little bit more downstream. And that can have a big impact on disease progression and managing certain diseases. All right, everyone, so I wanted to just give you a quick rundown of what biotechnology is. There is so much more to it than just that. I highly recommend, if you are interested in biology, especially molecular biology, biotechnology is a very, very exciting potential career path. As always, thank you so much for being here. Have a great week, everyone.